Hi, I'm Thomas, and I'll discuss the article Females Have a Different Metabolic Response to Critical Illness, Measured by Comprehensive Amino Acid Flux Analysis. Critical illness puts a huge strain on protein and amino acid metabolism. While men and women differ in hormones and muscle physiology, it's not well understood if they also differ in their metabolic response when critically ill. This study explores the differences by comparing male and female ICU patients using an advanced amino acid tracer method that measures how the body produces and breaks down amino acids in real time and identifies any significant specific metabolic patterns during critical illnesses, specifically focusing on whole body protein breakdown and how the illness severity influences these patterns. If males and females respond differently on a metabolic level, then using the same treatment strategies for everyone may not be optimal. For this study, the population included 51 ICU patients and 49 healthy controls for a total of 100 people. The researchers performed a secondary analysis using data from a previous phenotyping project. The original study only compared ICU patients to healthy controls, but this current secondary analysis study breaks down the data by sex and illness severity. A stable isotope pulse method was used where 18 labeled amino acids were given as a single IV bolus. Healthy subjects fasted overnight, while ICU patients had 2 feeds stopped for 6 hours. Blood was drawn from an artery in ICU patients and drawn from a heated hand vein in healthy controls to mimic arterial blood. Blood samples were collected over 2 hours to track how each tracer disappeared from circulation, and amino acids were identified by measuring the tracer's disappearance rate and how much of it reappeared as metabolic products. Using metabolic flux and compartmental modeling, they calculated whole body amino acid production, intracellular flux, and pathways of conversion. Protein breakdown was tracked through the conversion of phenylalanine to tyrosine pathway, a reliable indicator because phenylalanine cannot be synthesized in the body. This important pathway alters under metabolic stress, reflecting overall protein turnover. To summarize key metabolic concepts in the article, the body goes into a highly catabolic state with critical illness, causing amino acids to move through pathways involving glutamine, glutamate, glycine, and taurine, all supporting immune and oxidative functions. Citrulline is highlighted because it may reflect gut health. Low levels usually mean impaired enterocyte function. Different organs contribute in different ways. Muscle provides amino acids during breakdown. Liver handles most processing and urea cycle activity. The gut's citrulline output signals enterocyte function. The critically ill population had conditions like sepsis, high inflammation, and multi organ dysfunction with varying illness severity being assessed with the SOFA scores, which were divided into moderate and severe scores. This showed how metabolic patterns shift with increasing severity. Those with high inflammation were shown by high CRP levels. The study revealed females had larger increases in several amino acids, especially glutamine and glutamate, and also showed higher levels of taurine and glycine. When essential amino acids decreased, females tend to have smaller declines compared to males. This suggests that females maintain or elevate certain amino acid pathways more strongly during critical illness. The intracellular amino acid pools and metabolic fluxes were also more elevated in females, indicating higher metabolic activity at the cellular level. When illness became severe, females showed a sharper drop in amino acids like arginine, citrulline, histidine, and methionine. This pattern points toward metabolic suppression when stress becomes extreme. Severe illness also reduced phenylalanine to tyrosine conversion, reflecting lower protein turnover and reduced autophagy. Citrulline levels were significantly reduced, which signals impaired gut function, since citrulline is needed to produce arginine. Arginine pathways and nitric oxide production were also reduced. In contrast, taurine and glycine increased, not explained by protein breakdown alone. Overall, both sexes experienced high protein catabolism, but the metabolic response in females was more amplified, showing both stronger increases and more pronounced drops under severe stress. One new concept I learned from the article was the stable isotope pulse method. Instead of a continuous infusion, this approach uses a one time bolus of many labeled amino acids. which allows researchers to see whole body amino acid production with a lot more precision. Another concept was the compartmental modeling they used. The study divided metabolism into extracellular and intracellular pools and measured how amino acids moved between them. This gave a clear picture of metabolic activity that isn't visible from plasma concentrations alone. The third concept was how strongly sex specific metabolism affects amino acid kinetics. The article showed that females had greater increases in several amino acids, and yet, when illness became severe, they experienced more dramatic drops. These findings that can be applied in clinical nutrition for clinicians indicate protein and amino acid needs may need to be more individualized. The stronger metabolic shifts seen in females and the sharper drops at high illness severity suggest that nutrition support shouldn't always follow a one size fits all approach. Another clinical takeaway is the use of citrulline as a marker for gut function. Citrulline production drops during critical illness, possibly indicating impaired enterocyte activity, and guides decisions about and can guide decisions about enteral nutrition tolerance or other support. This also reinforces the importance of adjusting protein delivery during high catabolic phases and amino acid patterns when deciding on formulas or treatment. This explains why men and women sometimes show different trajectories in severity, recovery, and outcomes. For future research, the article points towards how sex hormones influence amino acid metabolism, whether females need different nutrition strategies at high severity, and how citrulline and arginine pathways might predict outcomes. There's also room to develop more sex specific critical care nutrition guidelines based on these metabolic differences. Thank you so much for your time.